Hello friends, today I am going to discuss the topic respiration. It is very important for any agriculture related exam. So let's see first slide. What is respiration? So respiration is simply breakdown of organic substance into CO2 and energy. Generally organic substances like glucose, this break down into CO2 and ultimately energy. So this is the simple breakdown of simply breakdown of organic substance into CO2 and energy. Now there are two terms, uh, catabolism and anabolism. So first of all, what is metabolism? Metabolism means all the physical and chemical activities which happens inside our body, which converts or use energy. So all the processes or activities which requires energy or which produces energy inside our body is called metabolism, like digestion, breathing, circulation of blood. So these all are metabolic process. Now the catabolism and anabolism. So catabolism, cata means something break down or tear down something. So catabolistic process are those process that tear down biomolecules. So process where biomolecules are broken down are called catabolism. Like respiration is a catabolistic process, and anabolism is a process that builds biomolecules. Like we can say that from amino acid polypeptide is formed inside our body. It is a anabolism. Or from glucose glycogen is formed. Glucose stored inside our body in the form of glycogen. From fatty acid triglyceride is formed. So these all are anabolism. Now the term slow oxidation and fast oxidation. So slow oxidation generally occurs at a low temperature, and it is catalytic in nature. That is, it involves enzyme uh, to happen, while the fast oxidation occurs at high temperature, and it is non-catalytic in nature. It doesn't involve any enzyme. So the best example I've given here, the burning is an example of fast oxidation, and the cellular respiration is a slow oxidation process. Basically, the chief product of respiration is energy output, and it can also be defined as slow oxidation of organic food. And the basic nature is exothermic. Its nature is exothermic, and the release of energy is due to the energy that is conserved in the biomolecules in the form of chemical energy. So in respiration, chemical energy chemical energy of biomolecules is utilized. So the we can say a simple equation of respiration as the carbohydrate or the hexose molecule. Hexose is with the help of enzyme converted into CO2 and H2O. And in respiration process, first of all, complex carbohydrate with the help of hydrolyzed by enzymes into hexose sugar, and then the cellular respiration of hexose sugar occurs. In case of fat, it is hydrolyzed by lipase into fatty acid. Then its slow oxidation occurs, and proteins are generally not utilized. It is utilized only at that time when there will be uh, no carbohydrate or fat available for utilizing. So now moving on to the next slide. So phases of respiration. Generally, respiration, we can say that it occurs in two phases. First phase of respiration, in where use of this use of carbohydrate. In first phase of respiration, carbohydrate is utilized. It is called floating respiration, and the name was suggested by Blackman. So this point should be noted that what is floating respiration, or the question may be asked like who named floating respiration? Then Blackman is the answer. And what is floating respiration? Then it is the utilization of carbohydrate, which is the first phase of respiration. So, and the second phase is the protoplasmic protein utilization. So, protoplasmic protein used called protoplasmic respiration. So, the name first one is the floating respiration, and the second one is the protoplasmic respiration. Now, the most common respiratory substrate. It may be asked that what is the most common respiratory substrate? So, the answer will be glucose. Glucose is the most common respiratory substrate. Now the types of respiration. So Sachs in 1890 he classified cellular respiration into two major types. That is, one is aerobic respiration and second one is anaerobic respiration. So aerobic respiration involves oxygen and anaerobic doesn't involve oxygen. So the first step or the first stage of first stage of respiration, in both cases, aerobic and anaerobic does not involve oxygen. But after the formation of pyruvic acid, which is under glycolysis process, after that, if oxygen is utilized, then the product will be CO2 and H2O, as shown here for the aerobic respiration. And for anaerobic respiration, the pyruvic acid without oxygen, it converted into ethyl alcohol, or in case of lactic acid fermentation, lactic acid is formed. So the main point to be noted here is that the energy released in case of aerobic and anaerobic respiration. In aerobic respiration, 686 kilocalorie of energy is released, while in anaerobic respiration, only 59 kilocalorie of energy is released. And this one in ethyl alcohol formation, and in lactic acid fermentation, only 36 kilocalorie of energy is released, while in aerobic it is 686 kilocalorie. So we can say that aerobic respiration is highly exothermic as compared to anaerobic respiration. So now moving on to the next slide. So there are basically four stages in cellular respiration. First one is the glycolysis process, and major point is that glycolysis occurs inside cytoplasm of our cell. So in cellular respiration, cytoplasm acts as the site for glycolysis, and glycolysis consists of 10-step process. And then the second process is pyruvate oxidation, which is a single step process where pyruvate molecule is oxidized in mitochondria. Then the third process is Krebs cycle. Krebs cycle is a 8-step process and it also occurs in mitochondria. And the fourth one, which occurs is a complex multi-step process, is the electron transport chain, which also occurs in mitochondria. So that's why mitochondria is called the powerhouse of the cell, because the major energy releasing process inside the cell occurs in mitochondria. So mitochondria is called the powerhouse of the cell. Now glycolysis. So as shown in this diagram, glycolysis starts with the glucose, and glucose molecule, which is firstly break down. First of all, the carbohydrate that we consume is converted into glucose molecule, which is a 6 carbon compound. And this 6 carbon glucose first processes with the help of hexokinase enzyme. Hexokinase enzyme, the phosphorylation of glucose occurs. That is, phosphate molecule is added to glucose. So here two phosphate molecule is added to glucose. So this is called phosphorylation. So I will give you a brief description of phosphorylation. So in phosphorylation, what happens that ADP ADP plus phosphate, with the help of 7 to 11 kilocalorie of energy, ATP is formed in phosphorylation. That is, phosphate is added. So phosphorylation may be light induced, or with the help of chemical bond energy, or with the help of oxidation energy. So when with the help of light phosphorylation occurs, then it is called photophosphorylation, which occurs in photosynthesis. And with the help of chemical bond energy, if phosphorylation occurs, then it is called trans or substrate phosphorylation, which is which occurs in Krebs cycle. And with oxidation energy, if phosphorylation occurs, then it is called oxidative phosphorylation. In whole ETS, electron transport system, this oxidative phosphorylation occurs. So after this, that is here, one phosphate molecule is two phosphate molecule is added from ATP. So 2 ADP is formed here, and here after this is formed, that is glucose 6 phosphate. Glucose 6 phosphate is formed, and with the help of isomerase, fructose 6 phosphate produces. With the help of isomerase here, fructose 6 phosphate is produced, and then fructose 6 phosphate, with the help of phosphoexokinase, again one ATP molecule, one ATP of energy is utilized here, and in presence of phosphoexokinase, fructose 1,6 diphosphate is formed. So here fructose 1,6 diphosphate is formed, and so in the first stage of glycolysis, loss of 2 ATP is there. One ATP is lost here, and one ATP is lost in forming glucose to glucose 6 phosphate, and one ATP is utilized in forming fructose 6 phosphate to fructose 1,6 diphosphate. And then the second phase of the glycolysis. First phase is the ATP consumption phase. So it is an endothermic process. And the second phase of glycolysis is the ATP production process, which is an exothermic process. So in second phase, the first phase product, that is fructose 1,6 diphosphate. Fructose 1,6 diphosphate, with the help of aldolase enzyme. Aldolase enzyme, it converted into three molecules of phosphoglyceraldehyde. This is, in short, it is written as PGAL. So three molecules of phosphoglyceraldehyde is formed. So three molecules of phosphoglyceraldehyde is formed, and this again, with the help of isomerase, is converted into dihydroacetone phosphate, and finally two molecules of dihydroacetone phosphate is formed, and this 3-PGAL, with the help of dehydrogenase, converted into 1,3 diphosphoglyceric acid, and again, and then there will be a release of energy in the form of ATP. The release of energy occurs, and finally we get pyruvic acid. So with with one molecules of PGAL, with one molecule of PGAL, that is phosphoglyceraldehyde, 2 ATP of energy is released, and one molecule of NADH2 is formed. This should be remembered that in glycolysis process, there are two molecules of PGAL which is formed in glycolysis. So for one molecule of PGAL, 2 ATP and one NADH2 is formed. So for two molecule of PGAL, there will be 4 ATP and 2 NADH2. This will be formed. So we can say the net product of glycolysis as 2 molecule of pyruvic acid, 2 molecule of NADH2, and 2 ATP. Why 2 ATP? Because 4 ATP is formed here in the second phase, and 2 ATP is utilized in first phase. So overall 2 ATP of energy is released. So this point should be noted that net product of glycolysis is 2 molecule of pyruvic acid, 2 molecule of NADH2, and 2 ATP. So this is all about glycolysis process. Now the process is pyruvate oxidation. So the end product of glycolysis was pyruvic acid, and this pyruvic acid, with the help of coenzyme A, there occurs a decarboxylation of this pyruvic acid, and in presence of pyruvic dehydrogenase, decarboxylation of pyruvic acid occurs, or pyruvate occurs, and the final product formed is acetyl coenzyme A. acetyl CoA. Acetyl coenzyme A is formed. This is called acetyl CoA here, and this acetyl CoA help in decarboxylation of carboxylic acid. Sorry, this coenzyme A helped in decarboxylation of carboxylic acid. So the pyruvate oxidation gives acetyl CoA as the main output. Now it causes the start of Krebs cycle. So Krebs cycle is named after the name of scientist, and it is also called citric acid cycle, as the first product of reaction is citric acid, and also called tricarboxylic acid cycle because the nature of organic acid produced in this reaction is tricarboxylic acid, so it is called like this. So what happens in Krebs cycle? First of all, acetyl CoA, this is formed from the pyruvate oxidation, gets here it gets converted into citric acid with the help of citric acid synthetase. Citric acid synthetase or citrate synthetase, it converted into citrate. Then with the help of aconitase enzyme, it converted into isocitrate. Then again here one here there will be isocitrate dehydrogenase. With the help of isocitrate dehydrogenase, alpha ketoglutarate is formed. So citrate is a 6 carbon compound citrate, and ketoglutarate is 5 carbon compound, and then succinyl CoA is formed after this step, and the succinyl CoA is 4 carbon compound, and then we can see from this diagram that fumarate is formed, and then malate, and the final one is the oxaloacetate, which again converted into citrate. So this is the whole process. It runs in a cyclic chain. So this that's why it is called a Krebs cycle, because it runs in a cyclic way. Now the point that is important for examination is that in TCA cycle or Krebs cycle, no oxygen is taken up. So the oxygen that is taken up in only in pyruvate oxidation process, and in that Krebs cycle no oxygen is required, and the total number of CO2 produced in Krebs cycle from two molecule of acetyl CoA is 4. So question may be asked like number of CO2 produced in Krebs cycle from two molecule of acetyl CoA, because in Krebs cycle two molecule of acetyl CoA is involved. So for one molecule of acetyl CoA, two carbon dioxide is produced. So for two molecule, 4 carbon dioxide is produced, and total number of hydrogen released will be 16 in Krebs cycle. 16 hydrogen will be released. So this is all about Krebs cycle, and let's talk about the energy which is released in each step of respiration, cellular respiration. So in glycolysis, 2 ATP of energy is released, as we have already seen, and in Krebs cycle only 2 ATP of energy is released, because it is released from FADH2, that is shown here, FADH2 in the form of FADH2. Uh, Through Krebs cycle 2 ATP of energy is released, and in electron transport system there will be 34 ATP of energy released. So this happens like in total respiration 10 molecules of NADH2 is released, because for oxidation of one molecule of NADH2 to NAD plus, 3 molecules of ATP is formed. So in this case we can see that for the whole electron transport system there will be 10 molecules of NADH2 released. So per molecule of NADH2, 3 ATP is formed. So for 10 molecule of NADH2, 30 ATP will be formed. And there are two molecules of FADH2 also formed. So in ETS system, so there will be 4 more ATP. So in overall ETS system, there will be 34 ATP energy produced, and so we can say that in glycolysis 2 ATP, in Krebs cycle 2 ATP, and ETS 34 ATP. So overall 38 ATP of energy is produced in the whole cellular respiration process. So now next slide. This is mentioned here how much energy is released in respiration. Now the efficiency of respiration. So when we talk about efficiency, so it generally means efficiency means uh, actual by theoretical. So in respiration efficiency we can say that actual energy to be released in respiration to the theoretical energy released. So uh, it is given that one molecule of ATP is equivalent to 7.6 kilocalorie of energy. So 1 ATP is equivalent to 7.6 kilocalorie. And we have seen here that 38 ATP of energy is released in respiration. So total energy released in respiration is 38 into 7.6, and the theoretical energy released that we have seen in the equation is 686 kilocalorie. So the efficiency of respiration will be 38 into 7.6 divided by 686. So it comes out to be 42%. So the efficiency of respiration is 42%. Now we can say that on the basis of site, on cytosol 2 ATP of energy is released because glycolysis occurs in cytosol, and in perimitochondrial space. In perimitochondrial space, 2 ATP of energy is released because the Krebs cycle occurs in perimitochondrial space. And in cytochrome, uh, 34 plus 2, that is 36 ATP of energy is released because the whole ETS system as well as the Krebs cycle occurs in cytochrome. So 2 ATP plus 34, that is 36 ATP of energy is released in cytochrome. And we can say that on the basis of phosphorylation, oxidative phosphorylation releases 34 ATP of energy, and the trans phosphorylation releases 4 ATP energy because as I have already discussed that trans phosphorylation occurs due to chemical bond energy, and chemical bond energy occurs in the energy chemical bond energy helped in phosphorylation in Krebs cycle as well as in glycolysis. So the trans phosphorylation involves 4 ATP release of energy in overall phosphorylation, and the oxidative phosphorylation occurs in the ETS system, so it will be 34 ATP. Now the term respiratory quotient or respiratory ratio. So respiratory quotient is the ratio of volume of released CO2 to the volume of absorbed O2. So we can define it as the respiratory quotient as volume of released CO2 divided by volume of absorbed O2. So if more oxygen is absorbed for respiration, then there will be less respiratory quotient. So if we are going to oxidize, or we are going to, if we are going with the highly reduced food for respiration, then there will be more oxygen that will be absorbed, so the respiratory quotient will be less than 1. For hexose as substrate, respiratory quotient will be 1. For highly reduced food, as I have already discussed, like fat and protein, respiratory quotient will be less than 1. That is for fat it is zero point 0.7, and for protein it is zero point 0.8 to zero point 0.9. And for highly oxidized food, less amount of oxygen should be absorbed for highly oxidized food. So for highly oxidized like organic food, like oxalic acid, malic acid, the RQ will be greater than 1. Now the factors that affects respiration. So it is basically subdivided into two parts: external factor and internal factor. Now the external factor is further divided into these factors, like first one is the temperature. Now temperature has a direct relation for respiration, and it is according to Van Hoff's law. For and it is valid for temperature range up to 0 to 45 degrees Celsius. And we can say that the Q10 value is 2 to 3 because for every 10-degree rise in temperature, the respiration rate increases 2 to 3 times. So Q10 value is 2 to 3, and the optimum temperature for respiration is 30 degrees Celsius. And light has uh, an important role to play in respiration because light increases the respirable material by increasing photosynthesis. So when photosynthesis increases, then more respirable material will be formed, more glucose will be formed, and meanwhile more respiration occurs. On the concentration of O2 in atmosphere and CO2 in atmosphere doesn't fluctuate a lot, so this factor is not uh, needed to be considered here. Water has a important role for respiration. So water shortage of water increases respiration rate. Generally, shortage of water increases respiration rate, but a very low concentration of water or content of water minimize respiration rate. Injury if occurs, then it will enhance the respiration rate, as we can see that in blemish portion of the fruits and vegetable, it respires faster, so it gets damaged earlier. So injury is directly related to the rate of respiration. And the chemical inhibitors like cyanides, azides, carbon monoxide, iodoacetate, chloroform, ether, this reduces rate of respiration. The mechanical effect like rubbing, bending, this increases respiration rate. High wind storm decreases respiration rate. And high wind and storm decreases respiration rate because it helps in closing stomata. So when stomata is closed, then uh, then the then there will be less respiration rate, or the respiration rate will get reduced because there will be more water concentration in the leaves. So water is inversely proportional to the respiration rate, and that's why the respiration rate decreases if stomata gets closed. Now the internal factors. Internal factor consist of protoplasmic factor and the concentration of respiratory material. The protoplasmic factor means that younger cell have more active protoplasm, so they respire rapidly than older cell. So it depends on the age of the plant or the cell, that younger cell will respire rapidly as compared to the older cell. And the concentration of respiratory material is an important factor, that if the respiratory material, that is the glucose or the other cellular material which are helpful in respiration, are more in concentration, then there will be more respiration rate. Now, the two important terms that are discussed here are the pasture effect and climacteric rise. So, pasture effect is generally, it represents increase in generally increase in oxygen, increases oxidation. This is what we have observed earlier. But in some cases, oxygen reduces the rate of sugar breakdown and even conserve it. And this effect is called pasture effect. And the climacteric rise, the production of ethylene in ripening fruit which increases respiration rate, is called climacteric rise. So generally this term is used to classify climacteric and non-climacteric fruits and fruits. So this is simply the production of ethylene in ripening fruit which enhances respiration rate. So this process is called climacteric rise. So that's all about this topic.
तो अगर आपको ये वीडियो अच्छा लगा तो प्लीज़ आप इसे लाइक कीजिए, यूट्यूब चैनल को आप सब्सक्राइब कीजिए और फेसबुक के पेज को आप ज़रूर से लाइक कीजिए ताकि हम जो भी नए वीडियोस डालें यूट्यूब पे आपको पता चल जाए कि हाँ हमने ये वीडियोस डाला, इस टॉपिक के वीडियोस डाले हैं। और ये जो चैनल है उसके बारे में आपको बता दूं कि ये यूट्यूब का चैनल एग्रीकल्चर फील्ड ऑफिसर IBPS उन स्टूडेंट्स के लिए जो एग्रीकल्चर से रिलेटेड किसी भी एग्जाम का प्रिपरेशन कर रहे हैं, जैसे कि IBPS का AFO है, नाबार्ड या फिर गवर्नमेंट की कोई भी वैकेंसी जिसमें कि एग्रीकल्चरल ग्रेजुएट्स का रिक्वायरमेंट है, उनके लिए है। और हम हर तरह के वीडियो इसमें डाल रहे हैं जिससे भी क्वेश्चन पूछे जा सकते हैं, जैसे कि एग्रीकल्चर के बहुत सारे टॉपिक्स और एग्रीकल्चर से रिलेटेड करंट अफेयर्स। तो आप लाइक कीजिए, सब्सक्राइब कीजिए और हाँ ज़रूर शेयर कीजिए ताकि इस चीज़ का फ़ायदा और लोग जो आपके साथ तैयारी करें वो लोग भी फ़ायदा उठा सकें। इसके अलावा जो एग्रीकल्चरल करंट अफेयर्स का जो एक सीरीज़ है, हर महीने में 2 से 4 वीडियोस में हम पूरा कवर करेंगे एग्रीकल्चर से रिलेटेड जो भी करंट अफेयर्स है, जो भी करंट टॉपिक्स हैं, जो भी न्यूज़ आ रहे हैं उसको हम कवर करेंगे 2 से 4 वीडियोज़ में। और इससे जो भी वीडियोज़ है इससे रिलेटेड लिंक अगर आप उस चीज़ को डिटेल से पढ़ना चाहते हैं तो वो वीडियो के नीचे में कमेंट बॉक्स में दिया हुआ रहेगा, आप डिटेल से पढ़ सकते हैं। और हाँ, आपको अगर किसी तरह का डाउट है, अगर किसी तरह का क्वेश्चन है, या तो एग्जाम को लेकर या फिर इस टॉपिक को लेकर या फिर कोई भी, आप ज़रूर कमेंट कीजिए। हम सारे कमेंट्स पढ़ते हैं और सारे कमेंट्स का आंसर हम ज़रूर करेंगे। थैंक यू वेरी मच।